I welcome you all on behalf of ISNM MISI webinar series. So this series is held every month, and it's going to be a 45-minute talk followed by a 10 to 15-minute question and answer session. And if you have any questions, please feel free to put it in the chat box, and then we'll take it at the end of the talk. Now, it's my absolute pleasure to introduce Dr. Gaurav Sharma, who is an assistant professor in the Department of Cardiovascular and Thoracic Surgery, with additional appointments in Advanced Imaging Research Center and the Department of Biomedical Engineering at UT Southwestern Medical Center in Dallas, Texas. His primary research focus is on understanding intermediary metabolism in cardiovascular and metabolic diseases. He's using stable isotope carbon-13 tracers coupled with NMR spectroscopy. He investigates metabolism in models of metabolic diseases and donor organs intended for transplantation. Additionally, his lab employs cutting-edge hyperpolarized carbon-13 MR imaging in patients with cardiovascular and metabolic diseases to enhance prognosis and clinical decision-making. Dr. Sharma serves on the editorial boards of multiple journals, including the Journal of Heart and Lung Transplantation, and he's a peer reviewer for NIH and other international funding agencies. Without further ado, Dr. Sharma.
Yeah, thank you, Sam, for the kind introduction. And let me begin with this talk. So today I will be talking about the mitochondrial metabolism of the cardiovascular diseases. My lab focuses on understanding mitochondrial metabolism, particularly in context of cardiovascular diseases, using advanced imaging and analytical methods like hyperpolarized carbon-13 magnetic resonance. And because this is an educational format, I will begin with the overview of the fundamentals of the metabolism and metabolic imaging. And let's begin with addressing one of the major issues in the field. So I would say there are the significant challenges that both researchers and clinicians are facing when dealing with the cardiovascular and metabolic diseases. And one of the key issues, if the patient has a diabetes, then the heart failure is present at least 25% of the time. And if the heart failure is present, then the patient has diabetes at least 40% of the time. And whereas if you see that the patient with the diabetes, then non-alcoholic fatty liver disease, and most recently the name changed to the MASLD, which is metabolic dysfunction associated steatotic liver disease, that is present at least 55% in the patient population. So what does it telling us? I mean, these are the comorbidities. And developing these comorbidities, you might see that it's kind of a long process and which include a very complex and integrate several things which goes together. And some of these, if you look at, for example, the obesity, hypertension, insulin resistance, hyperglycemia, dyslipidemia means the elevated triglyceride or reduced HDL. Together, they create a condition called the metabolic syndrome. So it's a syndrome, means it's the different diseases and comorbidities together creates a syndrome. And I won't be surprised, I mean, in a metabolic syndrome itself is present at least more than 1/3 of the adult population in the United States. And that is at least 1 in 3 adult. So this is a concerning topic. And I would like to point that there has been several advancement in the field, and the current understanding shows that how various factors intricately linked with the metabolic syndrome. For example, if you look at the cardiovascular diseases in 20th century, that's pretty much like myocardial injury, which is like hypertension, coronary artery disease, vascular heart disease, or the toxins. But if you look at the 21st century, the advancement in the field has advanced our knowledge with now it is more like insulin resistance, inflammation, oxidative stress. lipid deposit, and viruses. Recent pandemic is one of the great examples of the viruses. If we talk about the diabetes in the 20th century, it is seamless blood glucose and the insulin. But now we see that systemic inclusion of all the organs, for example, brain is involved for the metabolic regulation, and we see the temporal progression of the disease. For example, the neuronal control, oxidative stress, kidneys are included because it regulates the blood glucose levels and the insulin and absorption through the intestine. So several different organs are involved. It's not just the blood glucose and insulin now. So my lab studies some of the fundamental questions in the field. So number one question my lab is most interested in, what are the cellular and molecular mechanism of the various cardiovascular and metabolic diseases or syndrome? The next question we are most interested in is it possible to prevent and treat cardiovascular and metabolic diseases by manipulating enzyme pathways? And uh, and the third and one of the most important from the clinician's perspective, is it possible to image metabolic function and treatment response non-invasively in the cardiovascular and metabolic diseases in the patient? Because we wanted to image means it has to be the non-invasive and the technology should fit in the clinical workflow. So let me begin with some of the fundamentals in the field. I mean, I would like to set a good preamble so everybody understand what I'm talking about and give some fundamentals of the technology. So before I move forward, I just wanted to say topic is not just linked with the nuclear medicine, it's beyond that, but it is very important because we are addressing some of the important problems in the field. So one might ask, what is metabolism? So if you look at the metabolism, some of these pathways, for example, communication, growth, waste disposal, and they are attributing it to a definitive function. So basically, metabolism supports various biological functions and such as growth, reproduction, and it powers and regulates essential functions. So I would say in general term, if metabolism equals function, I would not be incorrect. So how to understand the metabolism? So we have several technology in the field, and one of the most important, which is predominantly suited to preclinical model and cell and isolated and the perfuse isolated model. That is metabolomics and the metabolic flux analysis. So what is metabolomics? So basically, as you might already understand that, it fits in a central dogma of molecular biology from genome to transcriptome to proteome. Metabolomics comes right before the phenotype. So it basically tells us what is happening. For example, if you look at two screenshots, that won't tell you what is the difference between the metabolomics and metabolic flux analysis. So in general, I would say metabolomics tells about what is happening. I mean, the static picture of a traffic. So imagine each of these car is a metabolite and you are taking a snapshot, which is telling us this is a metabolite. For example, the metabolic flux analysis, it's gonna tell us how it's happening. And if you look at these two pictures, and now I will tell, the difference is how these metabolites move and interact over the time. And you see that the flux, I mean, if you imagine these flux, I mean, between these roads, these are the different pathways, and these cars are the metabolites. So you can, if you have a sense of driving, you would better understand that what I'm talking about. So that's the difference between the metabolomics and metabolic flux analysis. And together they provide a comprehensive understanding of the disease mechanism. Now I wanted to give a quick overview of the metabolomics workflow. Like I mentioned in the previous slide about how the central dogma of the molecular biology fits in there. For example, genotype to genomics, transcriptomics, proteomics, metabolomics, fluxomics. This is basically considered as a relatively new domain, and the phenotype. So as you might see, the metabolomics and the fluxomics, they are the closer to the phenotype. Besides metabolomics and fluxomics, all of these tells us about what could be happen. And this metabolomics and the fluxomics tells about what has already happened. So typically, if you look at the metabolomics workflow, we design a study, for example, from the patient perspective, if I talk about healthy patient versus the patient with disease. So we do a sample collection. Typically, these are the tissue, blood, feces, or urine. And then we pre-treat these samples, means we do the metabolic extraction. Then we do the data acquisition, typically either by NMR spectroscopy or the mass spectrometry. Then after the data acquisition, the data interpretation, and the result validation comes, then there are sophisticated software tools which we utilize for the metabolic pathway analysis and the biomarker identification and the statistical analysis. And if we talk about the fluxomics, that's exactly the same, the next to the metabolomics, which is more closer to and provide a dynamic view of the metabolism. So we do the experimental design and we label with the stable isotope tracer, for example, the carbon-13, which is, very common stable isotope. And we know that the carbon is the backbone of carbon is the backbone of life. So you find that pretty much carbon is labeled with most of the important metabolites out there. So what we do is we do an isotope labeling, and then after the labeling experiment, we acquire the data either by the mass spectrometry or the NMR spectrometer. The advantage of using the MS is far more sensitive than NMR, and NMR is slightly more, comprehensive in terms of the data analysis. It provides much more information. So those are the few bigger difference between the two technologies. And we do the isotope tracer analysis, and after that we look at the dynamics and the rate of conversion by fitting in a different model. We do a statistical analysis and convert these flux with the metabolic model. So that's how these tells us the enzyme pathway is different, flux through the different enzyme pathways. So, but these technologies are great, can be applicable in the clinic. Well, like we give a tracer to the patient. These are relatively very safe, non-ionizing and non-radioactive, safe to use. But the problem here is we have to get a tissue to analyze, means the biopsy. It works well in the patients, for example, with the cancer. But in case of this, if there is a case we wanted to improve a diagnostic, we have to go non-invasive imaging. So we have a technology which pretty much work on the same principle, and definitely there is a need of another imaging method. So the question comes, why we need another imaging method, whereas we already have several methods available? So as you might see, the different level of the imaging at different, molecular level. You see that from genome to proteome and metabolome or metabolism, I would say. We need a molecular imaging technology. For example, genome, we need cell transcription, and the protein level, we need cell markers to image that. For example, if you wanted to image metabolism, the current standard is the PET imaging. PET imaging is great, And the most important probe is the FDG, which is most commonly being used. It is set as a current standard of the metabolic imaging, but it's most of the time it does not tell you the complete story. And the reason is the FDG or some of PET tracer, they just talk about the uptake. They do not further metabolize to provide comprehensive information about the flux through the various pathways. So, before I move forward with, anatomical imaging, which is kind of MRI, and the functional imaging with the vision. So, our interest in the metabolism, so I will talk about the metabolism. So we have a technology called hyperpolarized carbon-13 metabolic imaging. And one of the probe, what we currently use most commonly, that is called C1 label pyruvate. And this is carbon-13 label. For example, if you inject this in the patient, it rapidly goes in the bloodstream, go into the cells, and the pyruvate goes into the mitochondria, converts to acetyl coenzyme A, where it converts to the bicarbonate rapidly from CO2 to bicarbonate. And bicarbonate essentially is the marker of the aerobic metabolism. So think about this. You are imaging mitochondria, mitochondrial function, and all of that non-invasively. How fantastic it is. So and on top of it, it's not just about the mitochondria. You can get the information about the cellular redox as well. For example, pyruvate to lactate conversion typically considered as indirect marker of the cellular redox. So typically how data is analyzed, you take a ratios of the bicarbonate to lactate. Essentially, it is going to tell you about the bicarbonate means the aerobic metabolism over the anaerobic metabolism. So that way you get a comprehensive information about the flux through the various pathways and also whether it is sensitive to the mitochondrial metabolism or the cellular metabolism. But C1 label pyruvate is not just one probe. There are much more probe, and field has, while it is relatively new, but it has made significant strides in the field of imaging. And these are some of other tracers which is commonly used. And what you see in a purple color means the top four. They are already tested and approved at one of the site which studies the hyperpolarized carbon-13 metabolic imaging. And because this is human imaging, we we submit it for the FDA IND approval, and these four are already tested in the human. Rest everything which is currently posed for the clinical translation, and they also provide a different kind of information. So their potential clinical application on the right side of the screen. So all the different probes, they assess the different metabolic function, and it has several proven application. I would not go in the detail, but I would certainly give you a glimpse of it and with the references. Somebody wanted to take a look on there. So it can scan the cardiac ischemia and blood flow and perfusion, redox potential, and the pH in the tumors, and the treatment response, and the cellular transport rates. And these are just few examples. The scope is beyond these journal use, and you can probe the metabolism and onset of you need a dedicated probe which kind of include in that pathway what you are interested in, and these application, I mean, you can get more information about the pathways involved. But as I mentioned, the technology is relatively new, and if you look at the world map, it is only available at the handful of the institution. If you see that one of, UT Southwestern is one of the prime site. So overall in the world is there are almost more than 20 20 world site, which include the clinical polarizer And there are more than 15 clinical trials at current point of time, roughly about more than 20. So now I'm going to talk about some of the studies, of what we do and how we employ these methods. So if you look at these studies from left to right, cells and isolated perfused organs versus preclinical versus clinical, you see that from left to right, they become more relevant to the clinic if you go from left to right. But if you wanted to test a hypothesis and you have a limited money, then these models, cells and isolated perfused organs, they serve as a great method to test the hypothesis. But most of the hypothesis fails when they translate it to the clinic. So we work on each of the area. Our job is to translate some of the interesting finding. We first test in a perfuse isolated organ and then the preclinical model, depending on the hypothesis, and then we translate it to human. So I wanted to give you a few examples from each of that, and because we involve in the teaching, there are the computational teaching tools available for the students. So let's talk about the cells and isolated perfused organs. So here is one example. So one of our collaborator, Dr. Olson's lab, and they come up with this macropeptide, which is macropeptide regulator of the beta oxidation. So beta oxidation, basically every fat long chain fatty acid you consume, that basically goes to the beta oxidation. And this MOXIE is kind of micropeptide which regulates the beta oxidation. And they see that in the MOXIE it is conserved muscle enriched micropeptide, means it's if you look at the muscle, like for example the heart muscle, quad, and GP, you would see that it is more expressed in those tissue compared to any other tissue, for example kidney, liver, brain, and etc. And GAPDH is kind of a control. So it is more expressed there, and they find out the MOXIE is localized in the inner mitochondrial membrane. So if you see that the MOXIE flag versus the M-cherry, which basically localize it where it exactly located, it is actually located in the inner mitochondrial membrane. Then they have a question about whether MOXIE participate anything I mean, it involves in beta oxidation of the fatty acid, but what if, at what portion it regulates, and if we wanted to quantitatively analyze how this mechanism works. So basically, we design a stable isotope tracer studies using the carbon-13 label isotope. So here is one of two isotopes, which is glucose, which is labeled at the 1,6 position, and there's a long chain fatty acid. So I'm going to show you with the arrows. So if you look at this red arrow, which is labeled here, yes, and we analyze, once it metabolized, it goes inside the mitochondria, it goes into the TCA cycle, and what we analyze is the glutamate because it is relatively stable and it is directly linked with the TCA cycle, so it provides you very comprehensive information about it. So what you see coming from the glucose, you will see at the C4 label of glutamate, and it directly converts with the alpha ketoglutarate. And what you see from the long chain fatty acid, what you see in the green arrow, you would see that it is directly translated 4,5 label of the glutamate and the alpha ketoglutarate, and both of these are in the rapid conversion. And we look at these patterns. So these are the typical carbon-13 NMR spectra. And if you look at this, one need not to be an expert by saying that what are the gross changes among the spectra. So if you follow these arrows from the previous slides, the green arrows basically shows that what is coming from the long chain fatty acid, and the red arrows shows what is coming from the glucose. And you might see that this one, the long chain fatty acid, is kind of decreased. And if you calculate the fractional oxidation by sophisticated, the calculation, you see that the overall carbohydrate is increased and long chain fatty acid is significantly decreased, while there is no changes in the endogenous pool. And if you calculate the oxidation ratio between the carbohydrate and long chain fatty acid, that is significantly increase in a knockout of MOXIE. And so what does it telling us? It says the MOXIE plays an important role in the fatty acid oxidation, and it is required for maintaining energy homeostasis. Next example I would like to give is one of the, enzyme knockout, which is PKM1, pyruvate kinase muscle enzyme first. So if you have a sense of how the glycolysis pursues, so this is at the end point of the glycolysis, and you see that this activity is kind of very important, and pressure overload induced heart failure. And our collaborators, Wang, they look at the relative protein level of the PKM1 to the PDH, and they find that it's significantly decreased PKM1 activity in case of the heart failure. And when they look at the relative PKM activity, it's about the, it directly correlates with the PKM1 activity, and then they created the pressure overload model. This model is called TAC model, called as transfer aortic constriction. What they do is they basically tie a tag here, create a contraction. This is before tag and this is after tag. And then they look at the transthoracic echocardiography. And this is the sham, means these serves as a control, and they have sham knockout of the PKM1. Then they look at the tag of the flux, and both of these you clearly see that there is the contractile dysfunction in both tag model. However, by looking at these images, one can clearly tell, if you see at the knockout, there is a more contractile dysfunction compared to the control. So, and they also see the cardiac hypertrophic growth, which is adaptive response to the under the pressure overload. So we design another study. They are more interested in see that what happened to the glucose versus lactate pyruvate versus long chain fatty acid. So you see that the three different color pool, 1,6 label glucose, and we have a universally labeled long chain fatty acid, and we have a C2 labeled lactate and pyruvate. Because these have the same labeling, so we essentially cannot distinguish between the lactate and pyruvate. First, they are in a rapid conversion and they are similarly labeled. And if you look at these arrows, these are the one which kind of showing an increase compared to their knockout. So by analyzing these glutamate acetoprimers, we got the glucose oxidation. So in a flux, you see a significant increase in the glucose fraction or oxidation. And in case of knockout, you see a significant decrease in the glucose oxidation. Whereas the fatty acid oxidation, you see a significant decrease in both of the, knockout. Compared to the flux, they are controlled. Whereas the glycolytic flux, it follows the same trends as the glucose oxidation. And the TCA cycle flux, you will see an increase in the TCA cycle flux in the TAC model in both flux and knockout. However, the effect you see, the effect is more in case of flux, means they are controlled compared to the knockout. I wanted to give another example where we targeted the pyruvate dehydrogenase kinases. So basically, pyruvate dehydrogenase kinase is an enzyme which regulates the pyruvate dehydrogenase activity, one of the important enzyme in a right before the TCA cycle. And if you see that these are the diet-induced obese mice, basically we want to see that in case of the, if you look at their oil red O stain, this is diet-induced obese, which each of the red dot basically says that it has more lipid deposition inside the cells. And this is PS10, which is therapeutic intervention, and we wanted to compare it with the double knockout, means the genetic knockout. What happens if you therapeutically, you use therapeutic intervention versus the genetic knockout of it, which completely shut it down, the process. So we compare it. So by looking at the tracer analysis, we're using the same glucose and long chain fatty acid. We see that there is a decrease in both DKO and PS10, while where we see that the effect is more prominent in case of the DKO. And we look at the ketones in plasma, which is far more up in case of the DKO, whereas in PS10, the ketone body is much, almost the similar. Means the effect is more severe here. And we look at the cholesterol in the plasma. So we see that there is a significant decrease in the cholesterol in case of DKO, whereas you also see decrease in the cholesterol for the PS10, the therapeutic intervention group, but effect is much more severe in case of the DKO. So now I wanted to give you insight about the hyperpolarized carbon-13 workflow and the perfuse isolated organ. So basically, if you follow this arrow, basically it's going to tell you about the typical workflow. So we do a sample preparation. It can be a carbon-13 substrate and the free radical. Then we polarize it. We do a dissolution. Dissolution means we, give it a hot water and dissolve it. And after that, we do the carbon-13 data acquisition in a perfuse isolated organ. This is typical NMR system, spectrometer. We hang in there this perfuse isolated organ, which is directly perfused and connected to the roller pump and the perfusion setup. And while it is perfusing, we give a bolus of the hyperpolarized carbon-13 material after the polarization, and we get a very nice spectra, for example. So this is the same pyruvate which is converting to the lactate, and also through the acetyl coenzyme it is converting to the bicarbonate. You see that the pyruvate is a probe, and while soon after you inject, you see the signal is involved, and that there is definitely gradually decrease in that signal because of the loss of the polarization. And also you see that the downstream metabolites, soon after you inject, the downstream metabolites start appearing. So that is the real metabolism you barely see in any other technology. So here is an example where we gonna show that how we imaged redox. This is carbon-13 magnetic resonance spectroscopy. So basically the conversion of S to acetate to beta hydroxybutyrate that consider as a equation for the mitochondrial redox. And this basically translates by the enzyme called the beta hydroxybutyrate dehydrogenase. So by this equation, it is calculated as a mitochondrial redox, the ratio of the beta hydroxybutyrate to acetoacetate. So acetoacetate, we use the 1,3 label. So these two peaks are basically from the acetoacetate, and you see that all the conversion to the beta hydroxybutyrate and the acetyl carnitine. So by looking at the conversion of the beta hydroxybutyrate, you see that there are the four groups. Number one in control, then we created with the different version of the ischemia in the heart. This is global ischemia, means it's completely terminate the perfusion flow and creates ischemia for a long time. Low flow ischemia, where we decrease the flow about to 1/4. And rotenone is the chemical way of creating ischemia. It basically targets the mitochondrial ETS complex. And if you look at the area under the curve of the beta hydroxybutyrate compared to control, you see that the beta hydroxybutyrate is not produced in the global ischemia, low flow ischemia and the rotenone. If you look at the NADH, NAD plus NADH ratio, that is basically significantly decreased compared to their control. However, you also see the differences and more in case of the low flow ischemia because of the reperfusion compared to the rotenone group and the global ischemia. And I wanted to show another example where we utilized two different probes and for example, and imaged two different redox, cellular versus the mitochondrial redox. So if you look at here, this is the acetyl coenzyme this is acetoacetate, which is converted to the beta hydroxybutyrate, and this reaction serves as a readout of the mitochondrial redox. And whereas the reaction what you see in the blue, that is considered as a pyruvate to lactate conversion, considered as a cytosolic redox readout. When we see there's C1 of the lactate and C1 of the beta hydroxybutyrate, here are the some spectra about the same group, and here we use the AOA amino acetate. With basically what it does, it's basically blocks the flux between the cytosol and the mitochondria. And when we look at the metabolite ratio, though we see that there are uptrend in the low flow ischemia and routine on AOA group, but they do not reach to the significance level for the cytosolic redox. When we look at the mitochondrial redox, we see that there is significant difference in the low flow in the routine group. And we infer that this data is telling us the mitochondrial redox is much more sensitive to the cellular redox for these groups. And we published this history on the cover of the ACS sensor. And next, I want to give you a few examples of the preclinical and clinical study. Let's start with the translational study. I have already went through this HPMR workflow because here we want to scan a live animal. So the NMR is replaced from the previous workflow with the MRI system. We pretty much use the same, and typically, after the pH 7, we at the 37 degree, we give through the tail vein injection. And while getting the spectra, we typically look at the images in the preclinical model. And here's the heart what we are scanning. So here, we wanted to look at the myocardial response to the W-vitamine stimulation, and in a ischemic model in vivo. So this is typically the normal myocardium versus the ischemic myocardium, and you can clearly see the contractile dysfunction in case of ischemic myocardium. Here is the ejection fraction control versus what you call as the hibernating myocardium. And there is a decrease in the ejection fraction and the shortening fraction. We validate the model using the Dapian tunnel stain, and it clearly shows the evidence of the ischemia. Then we acquire time course spectra in vivo. And when we look at the normal versus ischemic myocardium, we do not apparently see much more difference in the bicarbonate, whereas slightly different, but in case of the W-vitamine, these difference are not significant. So if you look at, comparing, these are representative spectra, and when we combine all them together, you see that the normal myocardium versus the ischemic myocardium, you see a slightly, significant decrease, whereas you do not see the effect as anticipated in the normal myocardium versus the ischemic myocardium. And here is the bicarbonate to lactate ratio, which gonna tell us about the ratio of the aerobic versus anaerobic metabolism. And we see a bicarbonate to lactate ratio decrease. So why we are not seeing a difference in the normal myocardium and the ischemic myocardium with the W-vitamine? W-vitamine basically it mimics the effect of the exercise on the heart. So we look at the imaging and look at their 13C CSI maps, and after acquiring this in vivo. So this maps also, intensity map also confirm there is a decreased oxidative metabolism after the W-vitamine infusion. So we look at there could be a three possibility for these effect, which is kind of, not as anticipated. So one possibility is there is an increase in metabolism where you see that increase in the bicarbonate. Pyruvate goes in and then it converted to the bicarbonate. You would see the bicarbonate. The other possibility could be you might have fat and other substrate as a substrate competition, which might lower down the bicarbonate. And the third possibility is with the effect of W-vitamine, the heart rate increase and the clearance rate of the carbon-13 substrate also increase. So you might see that substrate clearing rate is also increased. Now I wanted to give you some example of the clinical history. So the hyperpolarized carbon-13 MRI is visible in the myocardium. So there is a report by the Chuck Cunningham in 2016. That was the first report which was published and this technology being translated to the human. He scanned four healthy subjects, and after that, a couple of years ago, this 2021 American Journal of Cardiology Imaging, they show that a group from the Oxford shows that the two patients with the myocardial infarction, they image this carbon 13 uh, of the heart in the patient with a myocardial infarction. So these are the anatomic images pyruvate, bicarbonate, and lactate, and you see that these are when these are overlay on top of the heart. You see that the pyruvate in the heart and the bicarbonate. That bicarbonate is typical. I mean, if you see a healthy patient, you see a typical donut-like ring, and it clearly shows the absence of signal means there is infraction in that area. Whereas the lactate, you see that ischemic myocardium is more used to the lactate utilization and production. So we are also interested in looking at the viable myocardium, that is the myocardium which can recover after the revascularization. So why it is important? So we all know the ischemic heart disease is the number one killer in the world. And if we talk about the viable myocardium, why it is important. So when there is a coronary artery disease, you see that typical normal flow in the heart, and when there is an abnormal blood flow due to the progressive atherosclerosis narrowing, you see that the regional wall motion and abnormality due to the chronic hyperperfusion. And then if you, by surgical intervention, for example, coronary artery bypass graft, or if you wanted to remove this plaque, you see they gradually return to the normal function and typically reach to the normal value. So why we are interested in imaging? So the randomized clinical trial, they use, it is not clear whether assessing the viable myocardium changed the outcome or not. This was published in Circulation 2016. That is why we see that this technology will certainly answer some of these questions, which is being unanswered so far. So here is the clinical 13C MR workflow at UT Southwestern. You see that because it is human, so more regulation and approval is needed. And all of this procedure are sterile. And we definitely need a clinical polarizer. So basically, our pharmacists first prepare these fluid paths. Fluid path is basically needed. These are the fluid path, and this is a sample vial which is loaded in this GE Spin Lab clinical polarizer. And there is a QC unit. Soon after the polarization, we test all these parameters whether they are within a range or not. Because this is a clinical polarizer, the major difference it can handle more volume and it can do 4 dissolution per hour, and it does not have any liquid helium consumption in it. So this is typically the workflow, and we have a dedicated MR technologist and the nurses who recruits the patient and take informed consent. Studies IRB approved, and after that we scan the patient after injecting the hyperpolarized carbon 13 with this matrite serum, and after the bolus. Soon after about 45 seconds, we start scanning the patient, and that produces really gorgeous images. So here's the grant which is recently funded by American Heart Association. So what we are trying to do here, I mean, this is kind of showing you the metabolism in the myocardium, the same using the C1 label pyruvate as a probe. And these are our clinical collaborator. Dr. Pels and Dr. Jason are the leading thoracic surgeon, and Craig Malloy is the leader in the field of the hyperpolarized carbon 13 technology and the medical director of advanced imaging. He is also a cardiologist. And you see that the three different groups. The group one is the healthy subject. Group two is the patient before CABG, and group three is the patient after CABG. So basically, we anticipate, and in case of CABG, we hypothesize there should be decrease in bicarbonate to lactate ratio because of the mitochondrial dysfunction. And after we revascularize using the CABG, we see the improvement in bicarbonate to lactate. And that's how we hypothesize. These are the equipment and setting for the human HP exam. And this is the typical Helmholtz loop coil, which basically this is from the pulse stack, which used to generate the images in our technology. So here is the HP13C exam, which we find feasible and safe and as in several patient. I'm just showing you one example from one of the patients. This is 78-year-old male, which is having a hypertension with a three-vessel coronary artery disease. This is the data from the left heart catheterization, and you might see that there is a blockage in the arteries. And we scan this patient preoperatively. The image you see from the bicarbonate for the heart and the lactate and the pyruvate, which is a probe. And we take the ratio of the bicarbonate to lactate, which is about 0.98. And post-operative exam, after the cabbage, we see that the right before the area which is showing the metabolic defect, it shows the increased bicarbonate in that region by looking at the arrow. So, and their bicarbonate to lactate ratios also increase. One of the interesting findings we see that the same patient also has the data from the FDG PET CT because the patient has low-grade follicular lymphoma. And we see that the bicarbonate and the lactate, and compared to the similar matching frame, we see that the FDG uptake is slightly more in exactly same region where it is showing the metabolic defect. So two different technology, what does it telling us? Does that provide a complementary information? Which is most likely the case because they work differently. However, I would like to mention that the patient is proposed, the preparation of the FDG pad for the cardiac scan is slightly different, means they been fasted compared, they been provided glucose for the preparation compared to the here. They are prepared differently. And with that, I just wanted to give a quick information about some of the computational teaching tools. And this is one of the examples. So we have a user-friendly computational tool on the left side of the screen, which is called the TCS sim. Basically, as the name suggests, it basically creates the simulation of the carbon-13 spectra. If you can check the metabolic model, what enzyme you wanted to see, then you can generate the spectra, how the spectra should look like if you have a different isotope moment. This is great for teaching and practicing isotope tracing and very useful in planning the new tracer studies with the experiment. Then we have a TCA calc. It's essentially, let's say if you acquire data from the mass spec or the NMR, you provide this, you load the data as the CSV and check out what is the labeling of the 13C substrate. It basically selects all of the 13 model and gonna tell you that which pathway would be a better fit, and you can calculate the flux through those relative flux through those pathways. So which is very important for analyzing the isotopic tracing data. And these are the great tool for practicing for the student, very comprehensive. Without even doing the actual experiment, you can get the anticipated whether your hypothesis is correct or not. So I reached to the final slide. So I want to talk about the final thoughts and takeaways. So disruption in the mitochondrial metabolism impair the cardiac energy homeostasis and contributing to the progression of the cardiovascular and the other metabolic disorder. We also learned the hyperpolarized carbon-13 magnetic resonance has the capability to image real-time non-invasive assessment of the mitochondrial metabolic fluxes and providing the clinical insight into substrate utilization in the heart. It is not just limited to the heart, it can provide the information of other organs too. The use of the HP13C can guide a personal treatment strategies by assessing the individual metabolic response to the therapeutic intervention, leading to the more effective management of the cardiovascular and metabolic diseases. And why I'm telling this, because the cardiometabolic health is different. For example, somebody who's sitting all the time in front of a computer and versus the marathon runner, you would see that the cardiometabolic health is different between the two. So you can guide a personalized therapy. So the treatment, for example, the person who is more active, who get these kind of intervention, the therapy should be different compared to somebody with a poor cardiometabolic health. And one of the most important thing I would like to emphasize, we all familiar with the leading journal called Nature. They usually publish, scientific research on top of their article. But I was intrigued by a few years ago when they published an article called "How to Collaborate" on top of the Nature article. So I would like to emphasize that if you really wanted to assess the metabolism in patient, that requires a strong collaboration between research and clinical teams. Without a successful collaboration, some of these studies would not lead a successful outcome. So I always advocate the team science. With this, I would like to thank our sponsor and funder, and definitely American Heart Association for several grants being fund. Most of these work are funded by the American Heart Association, and definitely my lab and the resources provided by the UT Southwestern, and our department chair, Dr. Michael Jason, who is very supportive, one of our collaborator, and Dr. Pelch, who is surgical director of the transplantation, UT Southwestern. And now wanted to, all of these, long list of collaborator, which is kind of making this work successful. So currently we are also translating some of these work, which is not we are not yet ready, but we got some interesting result in the lung as well. Some of my research is also linked with the transplantation, improving the heart and lung transplantation for end stage organ failure. So we have also making strides in that direction. If there are some students on the call, I would like to say that we have few positions. If you are interested in our work, you can certainly reach out to me. And finally, not the least, I would like to thank the organizer. This is a great platform. As more I'm learning about it, I see this is a great platform, and particularly connecting the both, experts from the US and also the young minds from the India who can learn about some of the cutting edge research in the United States. And with this, I would like to stop and open for questions.
Excellent talk, and I gained so much knowledge today, Dr. Sharma. Thank you so much for sharing your work and research and what you are doing at UT Southwestern. And I'll read out Dr. Halkar's comments. He's a professor of nuclear radiology at MD Nuclear Medicine. He said, great work from bench to bedside. For 4 decades, the only contrast that is extensively used with MR has been gadolinium DTPA, which just shows blood flow and third spacing. This C13 is a great new area.
Yeah, thank you so much, Dr. Holker.
Yeah, in nuclear medicine, with PET, we had used fatty acid with carbon 11. One of the problem with nuclear imaging is once the tracer that we give, if it gets into the metabolism, then we don't know where the signals are coming from. So that becomes a problem. So that's why F18 FTG has been the main workhorse, because it stops, it doesn't go further into the metabolism. C11 fatty acid went into the metabolism, and then even the same was with F-DOPA for brain work. It would get into the metabolism, then we would not know whether the signals we are seeing are from the DOPA or from the metabolites of the DOPA. So the image and the analysis became more and more complicated. So most of our advancement is trying to go to a fixed agent. The agent goes inside and doesn't get into the metabolism. It has its disadvantage. As you can say, it doesn't show the fluxomics as you refer to. It is still a static picture.
Well, it is fluxomics. The data I showed you, that's just one of the several picture we acquired. So we are currently developing a model which can provide you the signal over the time, similar to the images and the model I showed in the preclinical model, where you see that the signal is evolved over the time, For example, if you inject a pyruvate, you see a dynamic signal of the pyruvate from the several images, and that particularly, decrease. I wish I would have one of the slide which can show a different, uh, different images, how it evolved over the time. And typically, once you inject the pyruvate, you start seeing the bicarbonate and the lactate immediately. The signal first go up and then gradually goes down. So essentially, the people are developing an in vivo model, which they can calculate the KPL, means the pyruvate to lactate conversion, and the BP and the bicarbonate to the pyruvate. So they are, utilizing this applicable, but since the clinical translation is itself very challenging, so we are making, progress in that direction. So hopefully, when I will be invited next time, I will have some of the interesting data. But you have a very valid point, but about the carbon 11 acetate and some of the other which goes into the metabolism, but we have a question about the specificity. So
Yeah.
that is exactly one of the challenge we have been facing with the nuclear medicine. I would say, there is often a debate, one technology is superior to other. When my perspective is slightly different, I see that what works well for you. I mean, no one exam is kind of very specific. So I would say if you get two or more exam, future is multimodality. If you combine both of the exam, they provide a unique information about it. So you can leverage the information being provided by these non-invasive exam, integrate together and make a better clinically informed decisions. That's my
Um,
take.
I, I agree. There is no question about it, and ultimately, it is how well we can solve a clinical problem.
Yes,
Which tool doesn't matter as long as the clinical problem is solved.
exactly.
Um, for my nuclear medicine colleagues, long time ago, we did a test called C14 urea breath test
Right.
for
Yes.
H. pylori, and
Exactly.
then it was taken over by C13 by mass spectroscopy. I don't think now even that is very often used. Even I doubt it. There are um,
Yeah, and then
as
for the, people still have a scintillation counter. They use C14. It's still nowadays. I know it's one of the, I mean, all these technology, what do you see, whether it is PAD or stable isotope, they all based on the same concept, very old experiment with the C14. The reason why we use the C13, because in case of MR, if you look at the carbon 12, which is natural abundant, it does not produce any signal. It is NMR insensitive. That's why we go to the carbon 13. The advantage of using it, it is a stable isotope. It's non-radioactive, safe to use. So that's why, there's a lot of interest in the carbon 13, particularly. And why I'm most interested in this technology, because you can do same analytical principle. For example, NMR works on the same principle as MRI. So some of the finding, you test the hypothesis in the perfuse isolated organ of the preclinical model, you can translate it to the human using the same methodology or technology. So I understand you hit right on the money. Those old experiment is kind of basis of these
yeah.
kind of cutting edge work.
Yeah, and one advantage with carbon is probably it is in a micromolar concentration, so MRI signals can distinguish between uh, benign or normal and abnormal physiology versus pathology. The traces that are in nanomolar concentration probably that's the where MRI may not work very well.
I agree.
Or physiological functions that occur at nanomolar concentration may need radioisotopic techniques to distinguish between normal to abnormal.
Right.
Okay,
Um,
great. Thank you very much.
Yeah.
and thank you very much, Dr. Sharma. And I have shared your email address on the chat for our trainees and young scientists. If they are interested in joining your lab or some kind of research, they can reach out to you directly.
Yeah, 100%. I'm interested on, young mind who can help us taking some of these projects forward. And also, I'm open for collaboration if, like Dr. Hulker, he seemed, my apologies, I do not know much of the field in this, in the nuclear medicine, because I just, try to leverage both of these technology, but he seems like a leader in the field, kind of depth of knowledge he has. So I'm open for a collaborator and senior mentor in the field who wanted to collaborate and leverage some of these technology, how they integrate together and provide more informed decision and solve the bigger problem.
Sure.
Absolutely. Yeah, that's true. Dr. Halkar is very knowledgeable and has got very in-depth knowledge, and we can certainly talk offline and discuss some opportunities and see how we can collaborate.
The,
100%. And I see that this is a great platform, connecting the two different population from each country. And thanks to our organizer to keeping this program alive. I know a lot of efforts goes in the planning, programming and all. So thank you so much for working effortlessly in that direction.
Absolutely, you're most welcome. And since we host them on the first Saturday of each month, we'll what we do is we also send out invites to our all the previous speakers. So feel free
Yep.
to share it among your trainees, students, and if you want to join in the future webinars, you are more than welcome.
Thanks. I'm in. I'd love to hear some of the intriguing tech in this field. Thank you.
All right. So thank you everyone for your attendance, and this meeting is recorded, and those who were not able to attend, they can see the recording later. Thank you. Thank you very much, Dr. Sharma. Have a great day.
You too. Thank you. And happy Ganesh Chaturthi for those on call from India.
Yeah.
Thank you.
Ganesh Chaturthi.